Yes, we understand. Please press rozumiem. Okay, Aniruddha, please open your window, your camera.
And I will admit all now.
Yes, thank you very much. Dzień dobry Państwu. Otwieram posiedzenie Komisji do Spraw Doktoratów Rady Naukowej Instytutu Biochemii i Biofizyki Polskiej Akademii Nauk w Warszawie. Now let me switch to English because of the circumstances. So my name is Ewa Świeżelska and I have a pleasure to chair the first session of the Committee for Doctoral Thesis of the Scientific Council of the Institute of Biochemistry and Biophysics, Polish Academy of Sciences in Warsaw. The agenda of this meeting today includes public defense of the PhD dissertation submitted by Dr. Aniruddha Das. I would like to welcome you all, the committee members, all the reviewers and supervisor, and also all the guests who joined us today for this defense. Of course, most cordially, I would like to welcome Aniruddha, the main hero of the day. So,
thank you so much.
A few formal information. So we have 11 committee members with us today. We have also all the reviewers and the supervisor. So all the formal requirements to proceed further are fulfilled. Please keep in mind that the defense is being recorded. And then let me also remind you that the PhD procedure of Master of Science Aniruddha Das has been initiated on March the 3rd, 2020, and his project was performed in the International Institute of Molecular and Cellular Biology in Warsaw, under the supervision of Dr. Wojciech Pokrzywa. The title of the thesis is Mechanism and Function of Synergistic Cooperation of CHIP and UFD-2 ubiquitin ligases. There are three professors nominated as the reviewer of the thesis. These are Professor Krzysztof Liberek from the Laboratory of Protein Biochemistry, Intercollegiate Faculty of Biotechnology, University of Gdańsk. Good morning, Professor Liberek.
Good morning.
Dr. Itai Koren from the Mina and Everard Goodman Faculty of Life Sciences, Bar Ilan University, Ramat Gan, Israel. Good morning.
Good morning.
And Dr. Grzegorz Sumara, Dioscuri Center for Metabolic Diseases, Nański Institute of Experimental Biology, Polish Academy of Sciences in Warsaw. Good morning.
Hello.
So we have received the opinions of all the reviewers. They were positive, and they have suggested to proceed further with the promotion of the candidate. These opinions will be presented during this meeting. I am also obliged to tell you that Mr. Aniruddha Das has passed all the required exams, including the exam in biological sciences, with a very good result. So these were the formal data required for the Committee for Doctoral Thesis to allow for further steps of PhD procedure that has been also done in a special type of voting. And since all these requirements are ready to proceed further, we are continuing now with the public defense, and let me invite the supervisor to present the CV of the candidate. Wojtek, microphone is yours.
Okay, hello, can you hear me?
Yeah.
Because I don't see my screen. Okay. All right, so yeah, I wanted to say a few words about Aniruddha, who is basically my first PhD student, one of the first PhD students, and the first that probably is going to have a title. So I'm kind of proud of that. And yeah, I wanted to say a few words about him, about his work. So what I think and what's really true is that Aniruddha is a diligent researcher who has dedicated the last 5 years to pursuing his passion for studying the ubiquitin-proteasome system. So he puts really a countless of hours of hard work and effort to conduct research, analyze the data, and develop this thesis. So I think Aniruddha has demonstrated during this 5 years critical thinking, a lot of creativity, analytical skills, and that making him really a formidable candidate for the PhD title. So yeah, I think it's important also to say that I'm convinced that Aniruddha's successful defense of this doctoral thesis will be kind of a testament of his academic determination and kind of a promising career, because he will continue scientific career in a postdoc position in US just soon after the defense, further exploring the world of protein degradation. So and yeah, finally, I would like to thank the people from the Institute of Biochemistry and Biophysics of Polish Academy of Sciences, because they made a tremendous job in doctoral school and also organizing this event. And of course, the reviewers for their time. So Aniruddha, I wish you a successful defense, and I hope this will be also interesting for the audience. So yeah, good luck. Thank you.
Thank you, Wojtek.
Thank you very much, Wojtek. And now it's time for the candidate to present his achievements. So Aniruddha, please share your screen with us. Yeah, it works perfectly well. So please start your presentation. Okay, once again.
You can hear me?
Yeah, we can see
can,
your presentation. We can hear you very well. So please continue.
Okay. So good morning, everyone. I'm very glad to, I'll just put this laser pointer. Good morning, everyone. I'm very glad to welcome you all to my doctoral PhD thesis on mechanism and function of synergistic cooperation of CHIP and UFD-2 ubiquitin ligase. The key question: how cell can remove unwanted protein? It uses two main pathways: the autophagic pathway and the ubiquitin-proteasome system. Less commonly, protein can be expelled also outside the cell via vesicles. My work deal with the ubiquitin-proteasome system, or UPS, and my talk will be on this mechanism of E3 ligase activity regulation and physiological role. UPS regulates various cellular pathways through the removal of unwanted and damaged protein marked by ubiquitin-proteasome. So ubiquitin possesses a C-terminal glycine, which allows the formation of thioester bond with the sulfide group present in the catalytic cysteine of the E1 activating enzyme. And this is a ATP dependent reaction. After that, ubiquitin further transfer through a trans-thiolation reaction into the E2 conjugating enzyme. And later, this ubiquitin further transfer into the lysine of amino group of lysine of the substrate. Based on the E3 used in the system, transfer could be direct or indirect via intermediacy of thioester bond formation. And then further, after multiple cycle, polyubiquitin chain get attached with the substrate. And this reaction pathway can be reversed in the presence of deubiquitylase enzyme, which specifically recognize a specific ubiquitin chain. And once substrate get polyubiquitinated, they further get degraded by the proteasome, and the free ubiquitin further utilized another round of reaction. So ethyl ubiquitin ligases are responsible for both selecting the substrate for ubiquitination and mediating the ubiquitin marking with the substrate. Based on the ubiquitin transfer mechanism, there are different classes of E3 ligase enzyme exist. For example, HET E3 ligase can recruit the ubiquitin-conjugating E2 enzyme, and this ubiquitin get transferred to the substrate via formation of thioester intermediate because of the central cysteine residues present in the catalytic site. On the other hand, there are largest group of ET ligase exist known as ring ligase or really interesting gene, and cooling ring ET ligase are the largest member in this family. And this cooling ET ligase complex is consist of multiple subunits like substrate binding domain, adapter domain, as well as the ring box domain, which recruit the ubiquitin-conjugated E2 enzyme. And in this case, the ubiquitin get transferred directly into the lysine of the substrate. On the other hand, there is another class of enzyme which is used for specific class of degradation pathway known as U-box E3 ligase. They have exactly similar ring E3 ligase, only the difference is that they don't have the gene coordinates, and rather the U-box, the E2 enzyme binding domain is stabilized by the hydrogen bond and salt bridge. And the main feature, interesting feature of this U-box E3 ligase is that they can get self-ubiquitylate or auto-ubiquitylate. In that, because of that reason, when they recruit the ubiquitin-conjugating E2 enzyme, they simultaneously transfer their ubiquitin into the substrate as well as in the U-box E3 ligase. And the last class of E3 enzyme is known as ring between ring E3 ligase. They have the ring domain which recruit the ubiquitin-conjugating E2 enzyme, and ring 2 domain has the central cysteine residue, because of which it formed the intermediate thioester intermediate. And because of that reason, the ubiquitin get transferred sequentially from ring 2 domain to the substrate lysine residue. So when newly synthesized protein synthesized by the ribosome, they need to properly fold it in the 3-dimensional structure. And the protein quality control mechanism is actually important. Beyond this structural structure and function, chaperone need to play its role to properly fold the protein. And if protein are not folded efficiently, then they need to undergo a degradation via proteasome pathway. And this quality control, if fails, then they often form pathological aggregates. On the other hand, CHIP, which is a key quality control enzyme, it actually tilt the refolding and folding machinery of chaperone into the degradation pathway. And also, there are current and earlier reports suggesting that CHIP can also cooperate with different kind of classes of ET ligase to regulate specific substrate. But however, little is known about the molecular mechanism and functional role of CHIP-based ET complex. For the first time in MI et al. In Molecular Cell 2002, there is a publication showing for the first time that CHIP can interact with the RBR ET ligase Parkin, and this is which allow efficiently degrading the unfolded polar substance. And this is the first time showing that CHIP can cooperate with other ET ligase to efficiently degrade the substrate in the absence of HSP-70. And later in 2000, later it appeared that CHIP also can interact with the human ortholog of UB4B UFD-2 and involve in the regulation of UNC45. However, there is a controversy exists between the selectivity of the substrate. And also CHIP can cooperate with CF, another ET ligase, to regulate the eukaryotic initiation factor. Also, it can cooperate with MDM2 to efficiently regulate the aquaporin 2 as a substrate, and notch signaling known to efficiently degrade the TAL1 as a substrate. And this degradation pathway is efficiently regulated by the cooperation with SCF and the CHIP ET ligase. So CHIP may interact with other ubiquitin ligase, but the role of CHIP UFD-2 complex is poorly understood. It is interesting because the combination of ET ligase could support the formation of alternative ubiquitin structure and different set of substrate. Early C. elegans studies showed that CHN-1, the human ortholog of CHIP, can physically interact with the UFD-2 and allow efficient ubiquitylation of the myosin chaperone UNC45, thereby regulate the muscle development in C. elegans. However, there is a contradictory report showing that UNC45 is not a real substrate for this ET pair. So it's unclear how the high processivity of this E3 set is achieved and whether there are other specific substrates for this complex. To answer this, I studied the molecular function of CHN-1 and UFD-2 ubiquitin ligase complex. To understand and study the complex, it is important to first understand the structural aspect of the chip and UFD-2. Chip protein from different species form a homodimer, and this homodimer showed that it form a asymmetric. Why asymmetric? Because of the asymmetric deposition, only one of the U-box remain accessible for E2 enzyme binding, whereas the tricopeptide repeat region, which is accessible for efficient binding for the chaperone HSP-70 and 90. On the other hand, it's important to mention that UFD-2 is also a U-box domain containing protein, but in contrast to chip, it's a monomer. However, only a very few substrate of UFD-2 has been identified so far. Also, this protein first identified in yeast as E4 enzyme involved in UV fusion degradation pathway. First, we wanted to understand how is the high processivity of the E3 set is achieved. To answer this question, I carried out a number of analyses with recombinant protein to examine activity of this ligase alone and in pair. A quantitative assessment of an E3 ligase activity is generally performed by examining its auto-ubiquitylation in the absence of substrate. For this purpose, we perform a very simplified experiment using the recombinant protein of E1 ubiquitin in the presence of ATP and also in the presence of conjugating enzyme. So when all the essential ubiquitin component, ubiquitin pathway component are present, in the absence of substrate, E3 can get ubiquitolated. And that can be determined using a Western blot and probing the blot with the E3 specific antibody. And we observed that almost twofold increase in the CHN-1 auto-ubiquitilation in the presence of UFD-2 at a equimolar concentration. Additionally, I also concluded that the induction was unidirectional, as I did not detect any significant change in the auto-UV of UFD-2 in the presence of CHN-1. Further, I observed CHN-1 alone has a minimal activity, and I did not observe any change in the auto-ubiquitilation after approximately 80 minutes of reaction, whereas the change in the presence of UFD-2 is positively correlated with time. Next, to understand whether UFD-2 can regulate the processivity of CHN-1 independent of its E3 activity, just by binding to CHN-1, I used inactive UFD-2 variant, which is known as P951A variant. Data clearly shows that an enhancement of mono and polyubiquitination activity of CHN, regardless of the type of UV chain used in the reaction, when complex with inactive variant of UFD-2. Next, to understand the reason behind such increased processivity, I designed a very crucial experiment known as E2 discharging assay, which quantitatively assessed the cooperation of the ligase with the E2 enzyme by analyzing the increase in the level of E2 enzyme devoid of ubiquitin, which has transferred to the ligase. I observed that in the following assay that in the presence of inactive UFD-2, CH1 can discharge at least twice as much charged E2 compared with CH1 alone, which was revealed by the increasing accumulation of uncharged E2 as shown in the graph. This data indicate that UFD-2 binding to CH1 via the 3-pair domain enhance the cooperation between CH1 and E2, thus resulting in a more efficient E2 discharging and auto-UV. To further verify this possibility, I perform another auto-UV assay with the UB2W conjugating enzyme, which is known to maintain a strict 1:1 stoichiometry with a substrate and to catalyze mono-UV of chip. Considering this characteristic, I added CHN-1 and CH1 UFD-2 pair to ubiquitin charged UB2W and ubiquitin flat charged UB2W to follow the ubiquitination profile. When both E2 species are present included in the reaction, I should observe a mono UV of CHN1, but by UV and UV flat, and a twofold increase in the level of CH1 ubiquitination when complex with UFD2 compared with when CH1 is alone. And indeed, I observed that when I added this charged fraction of UB2W in the when CH1 alone and compared to when it is complex, it increased twofold increase of the auto-ubiquitilation of CHN-1. On the other hand, my earlier data showed that HSP-70 has a negative effect in auto-ubiquitilation, and also we observe in this assay that upon addition of HSP, even HSP-70, even in the presence of UFD-2, it decreased in twofold the ubiquitilation of CHN-1. Earlier studies showed that chip monomer form the most stable conformation when the extended helix of the HH domain support intramonomeric TPR U-box interaction, restricting the E2 binding surface of the U-box. Interestingly, UFD2 binding to monomeric CH1 did not stimulate the processivity. This data suggests that higher CH1 processivity upon UFD2 binding is achieved when both monomer are in dimerization state while disrupting TPR U-box interaction. Interaction of UFD2 should also bring consequence in the form of increased degradation of CH1 in vivo. As in vitro studies indicated that interaction with UFD2 enhance CH1 auto-ubiquitilation. Therefore, in vivo interaction of UFD2 could promote CH1 turnover. To verify this hypothesis, I analyzed the CH1 endogenous protein level in young adult worms, and I observed that in the absence of UFD2, there is significant increase of CH1 endogenous level in comparison to wild type worms, as well as the worms having inactive variant of UFD2. And also, I observed that upon proteasome elimination, it rescue the CH1 endogenous level, which suggesting that this regulation is happening through the UPS system, and UFD2 specifically involved in the regulating the turnover of the CH1. This data indicated finally that independent of ubiquitilation activity, UFD2 is involved in CH1 turnover, presumably by promoting CH1 auto-UB, which might lead to the proteasomal degradation. Hence, UFD2 here both an initiator of high chip ligase activity and a regulator of its appropriate level in animals. Interaction of UFD2 would stimulate the poly-UV activity of CH1, consequently leading to the efficient degradation of the specific substrate. To delineate the role of chip and UFD2 in vivo, I decided to identify such substrate. I searched for protein whose level increased after CH1 UFD2 and both ligase deletion to detect protein that accumulate in the deletion mutant. Level-free mass spec approach is used, and I observed almost 65 potential substrate which is regulated by this E3 pair, and this potential substrate shown to enrich in metabolic process and lipid biosynthesis. And this box plot analysis showing that ACY, one of the conserved S-adenosylhomocysteinase enzyme, which is actually increased their level in the absence of any of the E3 ligase. So S-adenosylhomocysteinase, S-adenosylmethionine is a main methyl donor for biological methylation modification. After the transfer of methyl group, SAM is converted to S-adenosylhomocysteine, the byproduct of methylation reaction and a potential inhibitor of methyl transferase. The only eukaryotic enzyme capable of SAH removal is ASCY, which catalyzes the reversible hydrolysis of SAH to homocysteine and adenosine. However, little is known about the regulation of ASCY. In East2 hybrid screen using the C. elegans cDNA library, we identified ASCY as a prominent interactor of CHN, and also I perform an immunoprecipitation assay using worm lysate, and it's suggesting that CHN can efficiently interact with the ASCY in vivo. And later I tested this by using the recombinant purified ASCY in the in vitro ubiquitilation assay. And I observed that when I added CHN-1 and UFD to both the E3 ligase in the system, it efficiently ubiquitilate ASCY as a substrate. And also it reflects in the auto-ubiquitilation profile. And to further validate this observation, I analyzed the polyubiquitinated ASCY fraction of wild type CH1 deleted and UFD2 deleted and CH1 overexpression line. We observed that there is a significant reduction of ubiquitinated fraction in the lysate where there is no CH1 or no UFD2. But when we overexpress CH1, we observe a significant amount of ubiquitinate fraction species, even higher compared to wild type animal. Next, we generated CRISPR-Cas9 knock-in GFP-based worm line to level ASUI used to track its localization and abundance without compromising the integrity of the worm while maintaining its natural expression level. And further crossed with CHN-1 null background, UFD-2 null background, and CHN-1 overexpressing background. And microscopic analysis of GFP ASUI fluorescence level revealed that a significant increase in CHN-1 and UFD-2 null allele worm, and a sub decrease when CHN-1 is overexpressed. To further understand the effect of CHN-1 and UFD-2 on the SAM cycle, I studied synthesis of phosphatidylcholine from phosphatidylethanol by phospholipid methylation pathway, which requires a significant amount of SAM and is sensitive to SAH level. In agreement with our assumption that deletion of either CHN-1 or UFD-2 would increase ASUI stability, leading to the intensification of SAM-dependent methylation via Saha hydrolysis and PEC conversion. Indeed, the ratio of PCP increase in CHN-1 and UFD-2 worms, which observed in our lipidomics analysis. The regulation of quality control ubiquitin ligase chip activity and its substrate selectivity is largely unclear. Here in this study, the extensive biochemical, proteomic, and lipidomic data unreveal the regulatory mechanism of chip processivity and reveal its role in lipid mechanism. Specifically, I managed to demonstrate here UFD-2 stabilize CHN-1 TPR and dimerization of the U-box domain, fostering an open conformation. HSP-70, on the other hand, leaching the U-box and TPR domain stabilize the closed state of the CHN-1, limiting the interaction with E2. And assembly of UFD-2 enable CHN-1 to regulate lipid metabolism via the ASUI. Interaction with UFD-2 also promotes CHN-1 turnover. So finally, I am very glad to let you know that this work appears as a cover highlight in EMBO journal. Here, UFD-2 portrays as a jasmine promoting the cooperation between CHN-1 and ubiquitin-conjugating enzyme, doubling the activity of CHN-1, which is represented as a double ball saxophone releasing ubiquitin. At the end, I would like to express my sincere gratitude towards IMCB, IBB, and all the members of Voitech lab, which make my journey wonderful. I would like to thank my thesis advocacy committee member and review committee member of this thesis, whose scientific input on my thesis are immensely important. I should mention that without generous funding support from FNP and NCN, this study won't be possible. Also, thanks to the core facility of CCAD and EMBL used for mass spec and lipidomics analysis was crucial. So finally, I'd like to thank you all for your attention.
Thank you very much, Aniruddha. Thank you for your nice, interesting presentation. And now, please stop sharing the screen. Dear candidates, dear members of the audience, we have been discussing your performance and the opinions of the reviewers and all the suggestions and comments. And after that, we have performed voting on acceptance of the public defense and on conferring Mr. Aniruddha Das, the PhD degree in natural sciences in the discipline biological sciences. And the good news is that all the committee members and all the reviewers voted for granting you the degree. So congratulations.
Thank you so much. Thank you. It's my pleasure.
The second good message of the day is that the second part of our discussion concerned the awarding of your thesis with a special award. And in this case, also the voting resulted in a positive result. So we will ask the Scientific Council for awarding of your thesis with a special award. So congratulations again.
Thank you so much. Thank you.
I should also admit that these are just our suggestions to the Scientific Council, and this is the right body to confer you the degree and the award. And this point will be raised on the coming meeting of the Scientific Council that is scheduled on March 28th. So pretty soon.
Okay.
So this is all from my side. And now microphone is yours. Would you like to add, to say something to us?
Yeah, so it was a wonderful journey from my side. Like when I came in Poland for the first time, I was excited as well as a little bit scared how it will start and all. And when I met my supervisor, it was a very exciting experience. He was very encouraging initially, and then with that encouragement, I started my journey, and we both working together to solve small problem, and then we could able to publish. And then all the lab member in the lab, it was like excellent group, and I have seen that how Wojtek lab is actually growing slowly, and lot of publication upcoming publication, and I wish I could stay, but I understand that if you want to learn more, then you have to go out from your comfort zone and start new journey. So overall, my experience being in Poland was excellent. It is a very crucial part of my life. I was away from family, but I still feel that I am at home, and I'm always focused in work, and Wojtek always pushing me beyond my horizon, and it was an excellent experience overall. And then finally, I wish I could prolong my stay and work more further, but yeah, as I mentioned, that if you want to learn more, you have to come out from your comfort zone and work ahead of your life. So yeah, that's all I have to say. And I would like to thank you again, all the people who joined this meeting, and mainly my supervisor Wojtek for his encouragement and full support throughout the tenure of my degree. And special thanks to Pankaj, who was actually working side by me. His contribution is very significant throughout my work, and he was giving me a lot of scientific input, and he always encouraged me to think beyond the horizon. And all the other lab members, they are also supporting in many ways, and there are other lab members who are also supporting in the establishment, experimenting, making plates, making reagent. All those things matters a lot for me. And again, I would like to thank every single person who was actually played a very important role for getting this degree. And I'm very happy to hear that I finally got this, and yeah, I have to look forward and move ahead. Yeah, I'm feeling a little bit emotional, but yeah, I'll be always keep my good feelings with me being in Poland. Yes.
Thank you so much, Aniruddha. Thank you for your warm words. It is a pleasure to hear that you are satisfied and so successful. Let me once again congratulate you and congratulate Wojtek, your supervisor. As Wojtek said, you are the first one to defend the thesis under his supervision, so it's a great day for you. Nice premiere. We wish you also the beautiful defenses of your candidate in the future. And now let me also thank all the people who worked on this meeting today. So let me thank the reviewers for your hard work. Let me thank the committee members for participating in this defense. And also thank you all the people who joined us today. This is a very important day for the candidate and also for the institute. Aniruddha, please take a look in the chat notes. You have congratulations for Vandana. Nice to read.